GMOs are genetically modified organisms that are genetically engineered animals or plants that have been artificially changed in their genes. This creates unnatural, unstable, artificial organisms such as plants, animals, and bacteria. They are created when scientists go through the process of GE, or genetic engineering. This process is when scientists select specific genes within one organism and insert them into a different species. Originally, GMOs were created to improve and benefit the species and other species that consume them. This is a fairly new technology. Examples of organisms that have GMOs are fiber and food harvests, which include corn, soy, canola, and salmon. It is said that GMOs inserted in these plants help the plant become drought tolerant, have increased yield, and defense against pests and viruses. GMOs are used for food products and are used for mass production of fruits, vegetables, and many other goods. Despite the scientists' claims that new organisms are tolerant to many harmful pests and pesticides, none of these GMOs are currently available to farmers and the public. They show no sign of tolerance, increased yield, and enhanced nutrition. Also, GMOs have been linked to health problems including allergies and also environmental damage. They also violate rights held by farmers and consumers. Are they safe? Most developed nations do not consider them to be. There have been many bans of restrictions in more than 60 countries on productions and sales of GMOs. Some include Australia, Japan, and the entire Europe continent. However, the U.S. has approved them by studies conducted by large corporations who create them and profit by them. However, many Americans choose to opt out of the GMO products. Many people fear the risk that genetically modified organisms may have unforeseen health risks and believe they are unsafe. GMOs have allergy-producing genes, and because companies do not label their products, it is possible that someone may have a fatal allergic reaction. However, none are evident currently, but can arise in the future. Because GMO technology is fairly new, there is a small amount of research which has been conducted on the long-term or short-term effects on humans. The greatest danger lies in the effects that cannot be anticipated yet. Environmental risks. A major concern involves transgenes or engineered genes being spread throughout the environment by reproduction. This isn't positive because it can cause an increased use of more chemical use and other harmful human products. Effects on untargeted species are also common. For example, Bt corn, which produces its own pesticide, is in use today. Concerns have been raised regarding adverse effects on monarch butterfly populations because of this pesticide. Labeling of GMO in foods. One of the most common controversial topics with genetically modifying foods is the labeling of the end products. A majority of people living in the U.S. believe they should know if GMOs are evident in what they eat, while the rest of the U.S. do not seem to care. People are unable to tell if the products they purchase are genetically engineered because there are no food labels on products that specify if they contain GMOs. You cannot tell if a physical difference between the foods, and corporate companies choose to leave the labels off because they are not required by law.
The term cloning applies to a process known as somatic cell nuclear transfer. For example, a cell from an adult cow, a donor, is extracted from one cow and inserted into an egg cell from another cow. The egg cell has had its nucleus removed so that it will read and duplicate the DNA from the donor cell. The newly created embryo is then zapped with electricity so that it starts multiplying until it becomes a metablastocyst, which is a small clump of cells that forms after an egg is fertilized. It results in a newborn that is identical to the donor cow. This is done for many different reasons. One reason is animal models are often genetically engineered to carry disease-causing mutations in their genes. As a result, cloning could help reduce the time needed to make a transgenic animal model and the result would be a population of genetically identical animals for a study. Also, genetically identical stem cells are sometimes used for medical purposes, such as growing organs. Stem cells that are cloned from organisms with a disease could be studied to help researchers develop treatments for that disease. However, problems with cloning animals through somatic cell nuclear transfer is simply inefficient. The success rate ranges from 0.1% to 3%, meaning that for every 1,000 tries, only 1 to 30 clones are actually made.
Cloning plants. Nature has been cloning organisms for billions of years. For example, when a strawberry plant sends out a runner, a form of modified stem, a new plant grows where the runner takes root. That new plant is a clone. Similar cloning occurs in grass, potatoes, and onions. People have been cloning plants in one way or another for thousands of years. For example, when you take a leaf cutting from a plant and grow it into a new plant, vegetative propagation, you are cloning the original plant because the new plant has the same genetic makeup as the donor plant. Cloning plant DNA is a key step for genetic engineering, gene studies, and other applications in plant research, both in discovery and applied settings. Plant biotechnology continues to create new opportunities in agriculture, environmental biology, chemical production, and medicine. Legal and policy issues. Cloning allows growers to mass produce plants that may be difficult to grow from seed. All the plants are genetically identical, which is useful because you can be sure to see their characteristics. On the other hand, the lack of genetic variation means that if the plants become exposed to disease or to changes in environmental conditions, all of them will be affected.
Genetic engineering is the process of manually adding new DNA to an organism. The goal is to add one or more new traits that are not already found in that organism. It involves taking genes from one species and inserting them into another. It is a common term for recombinant DNA technology. For example, genes from an Arctic flounder, which has an antifreeze property, may be spliced into a tomato to prevent frost damage. In humans, designer genes that are genes altered or created by genetic engineering are used for medical treatment or for research purposes such as preventing or curing disease. Ongoing advances make it increasingly likely that scientists will someday be able to genetically engineer humans to possess certain desired traits. Genetic engineering has the power to improve everyone's quality of life. It will soon be possible to improve our mental, physical, and emotional capabilities, as well as being able to introduce regenerative functions natural in other animals, increasing longevity, and ensuring a healthy diversity. Genetic engineering carries a promise of enabling a wider range of long-term survival. Not all human genetic engineering is targeted at improving future generations. In fact, human genetic engineering today, for all intents and purposes, is limited to gene therapy, which is the correcting of genetically defective cells in a single living human. Human germline genetic engineering refers to the altering of DNA in germ cells that will lead to the change of the reproductive cycle, passing the improved DNA from one generation to the next, while gene therapy targets not only the specific cells that contains DNA, but the specific strand of DNA that is defective. However, the possibility of human genetic engineering raises numerous ethical and legal questions. Gene therapy is very unsuccessful. Most scientists say that the process will itself generate new mutations that will be passed on to future generations.